So it is going to be a bit tough to interact with people, I guess, because so for everyone that is listening, I guess you are connecting to the Zoom panel via the platform of the meeting. So as you know, you can ask questions there. There is a discussion forum, and we can also open some polls for raising questions, etc. So François started one poll right now, asking you if you already have the environment to work on this tutorial already set up or not. So just to see how many people is interacting, etc. Can you quickly vote? Because probably we can spend a few minutes explaining how to do this. Yeah, okay. I'm going to share my screen. I think it will be more useful. And I think we can start. Okay, so there are still some people working on the environment. It went okay, apparently. So what I'm going to do is quickly reproduce for the ones that had trouble or couldn't make it yet. I can quickly explain how to install all this. So you should be seeing now my screen. And there are some recipes here on how to install the environment. Basically, I mean, if you're familiar with all these tools, it's very easy. You just need Git. It is important that you create an environment with Python 3.6 because this is more compatible with the tools that we are going to be using. And very importantly, you need to update ETE to the latest version, 3.1.2 with pip, preferably. You also need to install ETE tool change from the ETE Toolkit Conda channel and have some Jupyter notebook. So I'm seeing that many of you are already working on it and most of you already have it. So I'm going to wait until everybody has environment. Yes, thank you, Robert. So the link I posted in the discussion forum because I didn't know where to put it. So it's in the GitHub of ETE Toolkit. So github.com ETE Toolkit, there is a repository called cores. So you can enter there and you should see this this repository. And here is the whole instructions. Yeah, so we can, I think the discussion forum is probably the best way to interact with you. There is, so we can chat here in this forum, but I'm going to be explaining the tutorial and the exercises together with François. If there is some question relevant to the things that we are explaining, you can also raise a question and it will pop up for us and we can read the question and explain for everybody else if you want. So is anybody having problems installing the environment? If so, you can tell us here in the chat. And in the meantime, I think I can start explaining a little bit the idea of this tutorial. So this is going to be weird for everybody, I think, because we were thinking on a full day tutorial interacting with people, with exercises where you can work on and with more time, etc. So we have to switch everything to a 3 hours webinar where the interaction is, as you see now, is not ideal. But we didn't want to reduce the topics that we wanted to cover. So we are going to try to cover all the topics that we were planning. And for this, what I'm going to do is to work on the notebooks that we have prepared showing you how to solve the different questions in a blank notebook. And then you can try to do it at the same time as we are doing. And if there are questions, we can try to solve it, but very quickly. So we will, so the idea is that this tutorial is not going to be like any other documentation that we have for the ETE Toolkit. So I guess most of you are familiar with the ETE Toolkit if you are in this tutorial. So the ETE Toolkit is a package, a Python package that allows you to interact with phylogenetic trees. And we have documentation, technical documentation for the ETE at different layers. So we have the ETE tutorial, which is technical documentation, but still guided in different sections, etc. We have the reference guide for the ETE API. So you have the description of the different functions, etc. We also have documentation for the command line tools that are included in the package, and some cookbooks that are also explaining how to use the tools, etc. But none of those materials are actually covering ETE from a biological perspective. So if you really want to do some phylogenomic analysis, how you use ETE, and this is what we are trying to address with this tutorial. So what we have prepared is basically an exercise. You have the title and description of the exercise here. It's an exercise where we have fabricated some biological data set. Is fake, so never use it for real research, because we modified some stuff to get the results that we wanted, and where we are setting the following scenario. So imagine that you are after many years of work, your lab has just isolated and sequenced a very interesting strain of a bacterium called Aquifax aeolicus, sorry. And your strain is remarkably interesting because it possesses some remarkable resistance for sulfur-rich environments. So imagine this is of course fake. So we have isolated and sequenced this genome, and now you want to address a phylogenomic analysis of this genome. So what you would do in a real scenario would be, okay, so let's grab other genomes from reference species, and particularly other species that are also involved in sulfur-rich environments. Let's put all these genomes together in a FASTA file, for instance, and let's try to analyze it from an evolutionary point of view, okay? In this exercise, what we have compiled is a set of 15 species where you have one tax ID that is invented, this one, Aquifax aeolicus new strain. This is your strain, so this tax ID is actually invented. And then you have the actual reference genome of the Aquifax genome in RefSec, and some other bacteria and archaea genomes. And we have put all this into a FASTA file, and now we want to address a phylogenomic analysis from many points of view. And each section, I mean, each of the analysis that we are going to be showing you, oops, okay, reload. So each section that we have prepared is bound to a notebook where we are working on different features of the ETE Toolkit, addressing different biological questions. And there are two of them that we are going to skip because they are not related with ETE, and they are basically just explaining how would you generate gene families and the FASTA files for these gene families, so you can start working on phylogenies, etc. Anyways, I'm going to go very quickly over these two notebooks to explain some basics. So I guess you are seeing my screen with the two browsers, and if the text size is not good or whatever, just let us know and we can increase it. So I'm going to open, and you can try to do the same as I'm doing, okay? And try to reproduce. So I'm on the right, I'm going to open the notebooks that contain the solutions and the explanations. And on the left, I'm going to hack a little bit with the data. So the first notebook is just some explanation. So I just want to mention for you all, if you are interested in doing some phylogenomics, and especially if you are planning to use ETE, that there are some practical advices that you should consider regarding how you format the data. And these are the things that we took into account when we prepared the data that you are going to be using today. So first of all, save all your sequences as a FASTA file. This is in principle more convenient. Verify that the new line characters are consistent and preferably in the Unix form. Verify that all sequences are also consistent in terms of symbols and residues and etc. So they do not contain stars and weird symbols, etc. Also avoid long FASTA headers. So we want short sequence names. Do not, for convenience, do not wrap long sequences into several lines. This is, I mean, of course you can do it, but for some hackings that we usually do in the command line, is better if you have something like this, where you have one line with the name in the FASTA file, and then the second line, the next line is the sequence, all in the same line, so it is not a split. So this allows better grepping, counting, etc. Use, very important, use the same format for all the sequences, all the sequence names. Avoid symbols that conflict with the NIWIC format. So the NIWIC format is the format used for storing phylogenetic trees. So if you want to do phylogenomics and you are going to be generating a lot of phylogenetic trees, avoid having all these symbols in your sequence names, because this is going to crash many programs. Also make sure that there are no duplicates in your data set.
Can you increase a bit the font size?
Here. Let's try like this. Yeah, make sure there are no duplicate names. Make sure that, and this is very important for ET related analysis, make sure that the species code associated to each sequence name is encoded in the same, in the name itself. This is a common way of doing it, but if you do it in a very special way, ET will understand this much better. So the format that we are using in this exercise is always the species code dot and then the sequence name. And also the species code that we are using is always the NCBI tax IDs. So if you do this in the first place, before starting any analysis, then you are facilitating a lot of analysis later on. You don't have to do anything here because we already did. And if you go to the, sorry, where am I? Here. If you go to the data folder in the repository that you downloaded, these all prods, FASTA and FASTA nucleotides include all the sequences and sequence name formatted in that way. And this is what we are going to be using. So let's start with the first step to any phylogenomic analysis, which is we have a bunch of genomes. We need to organize the sequences into families. Okay, yeah, François is talking on a different screen, sorry. So we first need to generate gene families, I was saying. So this is grouping sequences by homology. And this is essential to do any phylogenetic analysis and also any phylogenomic study, because you need to organize the sequences by an evolutionary relationship. In this case is what I'm referring by homology. And to do this, there are many different ways, and we are not going to do it here because it takes a little bit of time that we don't need it, and it doesn't require ET. So this you can do it as you want. What I'm going to show you is how we build the data that we are going to be using here. So if you open the first notebook folder, building gene phylogenies, and you open the notebook, here you go. So you don't need to execute this, and I'm not going to execute it. I just want to explain to you how we did it. So in this case, we use a BLAST-like clustering program that is called MM6-2. There is actually a tutorial running tomorrow about MM6 and related tools. And what we are doing here is basically we take our files, our input files, all proteins, all prots in amino acid format. We create a database with mmseq, like in BLAST. We cluster all the sequences in database with this command, mmseq Linux cluster, with some parameters that this is controlling how similar the sequences should be, etc. And we convert the result of this clustering into a tab delimited file that is called this all prods cluster folder, blah, blah, blah. We don't need this. We have done this for you. Then from this clustering, we are going to generate a FASTA file for each family. And this is what we are doing here. I'm just explaining this for you if you want to reproduce this later, etc. But we are not going to actually run it. So this is the code that you could use to generate the FASTA files. It's using some features of ETE, but you could do it in any other way. Basically, we are loading all protein sequences into a dictionary, an ETE dictionary that is called seqgroup, and all genes in a different dictionary. Then we can process the file containing the information. Do you see my pointer? Francois, can they see my pointer? I guess so.
I see it.
Okay. So then you can read the file containing the information of the clustering, and then retrieve the sequences, the amino acid and the nucleotide sequences of each cluster, and generate a FASTA file. And this is what we are doing here. You don't need to do it because this is not involved this is not related to ETE, and we are providing the data. But here you have all the information. So now let's move to the next notebook, which is now we have the FASTA files. So wrapping up where we are, we have the 15 genomes, including our problem genome, and we want to analyze it from a phylogenomic point of view. We generated families out of those genomes, and we have a FASTA file for each family. So now for each FASTA file, we want to reconstruct a phylogeny, and this is what we can start doing using ETE. So how we reconstruct phylogenetic trees. So most of you, I'm going to make this a bit larger for now. So most of you are probably familiar with the phylogenetic workflow, the normal phylogenetic workflow. So what you usually need to reconstruct a phylogenetic tree is you have the sequences, and not aligned sequences, just unaligned sequences. You align them, you create a multiple sequence alignment, and then you can infer a tree. And in between these two steps, you can also do like typical steps are alignment trimming. You want to clean the alignment or remove gappy columns, etc. And in some cases, you also want to include some model testing steps. So this basically ETE considers these 4 general steps: building the multiple sequence alignment, trimming the alignment, model testing, and inferring the tree. And then ETE provides a tool, a command line tool that is called ETE3 build, that you can call from the command line, where you can use to workflow all these 4 steps in a very easy way. So this is what is explained here. There is a lot of technical documentation about how to use this. You have the cookbooks and examples in other places, but for now, we are going to focus more on the analysis rather than explaining all the options in ETE. So all you need to know is that if you call in the command line ETE3 build with the input file, one of your FASTA file, and then you use the -w parameter to define the workflow, then this workflow name automatically is creating the workflow. So here I'm saying, because ETE build has binding to many programs, and you install all those programs along with the installation process of ETE. So you have bindings for MAFT and tCoffee and D-Align TX and Muscle, etc. for aligners, Trimal for trimmers, model test for model testing, and RAXML, IQ3, FAST3, etc. for tree inference. And there are some default configurations for each of those. So you can define the workflow very easy like this, and then input file, workflow that you want to use, and magically you get your tree. So we are sorry. Oh no, I don't want to move this. I want to move this. Sorry, I have two windows. Yes. So now we can start playing with our data. You have all the examples here. I'm not going to just press enter and see what happens. I'm going to start working on our notebook. So I open a clean notebook, empty one. I just copied the description of the exercise, and we can start working here. So first of all, make sure that you have access to the folder data here. Yes, we have. And make sure also that you uncompressed the tar files that were included in data. When you downloaded the repository, we included this. So these are the results coming from the previous notebooks. Okay, so these are the phylogenetic trees that we generated and the FASTA files, etc. So we have access. Good. So now, if we call ET3 build, what we should get is just the help of how to use ET build. And as I said, we can infer phylogenes very easily with ET build. We just need to provide a FASTA file. So we pick one of our FASTA files, random one. So you have in data, you have the FASTA folder, right? So we can access any file, any amino acid file that is there, and use it as input. And then I want to create a phylogenetic tree. So I'm going to use a workflow that is MAFT, default for the aligner. Then I don't want any trimming here, none. I don't want any model testing for now, and I want to build a very quick tree. So I'm going to use fast tree default. I can make this larger. So if we do it like this, we are done. We just need to provide an output folder. So test tree. This is the output folder where our tree is going to be reconstructed. I mistyped this default. Here as well. What is happening?
And T
first default. It was a typo there. Okay, now it's running. So you see now that ET build is running, the first thing it's doing is checking that all the tools associated are okay. They are detected as installed, and then it starts concatenating steps. So the first thing it does is loading the FASTA file as a multi-sec task. Then the alignment. So is registering an alignment task that is actually using 15 amino acid sequences detected in the FASTA file and the MAFT program with the configuration that we decided. And then after this is running is inferring a tree with FAST tree. And then the output is here. So we have here already our phylogenetic tree, as easy as it. One command line, and we have the tree. We can actually, oops, sorry. Okay, now you notice that I try to re-execute this. It is not working because it detects that there is already an output. You can actually use the resume, and then nothing is going to be computed. It's just checking. Or if you want to clean up all the folder, you can use the clear all parameter. Okay, we don't need to do this. So resume to make it faster. So it's just checking that the files are there. And now we are going to see what is inside test tree. So when we list the output folder, what we have is a DB folder, which is internal, some login, etc. tasks is also internal. The folders that we are interested here are the MAFT, default, blah, 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 etc. Because this is representing the results for this workflow. And here, and if you, so here we have the alignment that it was used, the NIWIC, the tree that we obtained, even images that it created by default, the default workflow, and in different formats, and the alignment that it was used. So now, what else can we do with ET build? So now with one command line, we can generate one phylogeny. So all we need to do is to automate this for all our input FASTA files. But we can do actually more. We don't need it for this exercise, but you can run ET build with multiple workflows, etc. I forgot to mention that there's another handy tool, ET view, that you can use to from the command line also to inspect trees. So you can call ET view and provide a phylogenetic tree here. So the full path is this one. Oops, copy. You see that the pop-up. So this is by default opening the graphical user interface of ET that is also included in the package with the tree that we just generated, right? And I think in the notebook, there are also some cool examples where you can use the ET view command to quickly view the tree, but also the alignment that it was used to generate the tree. So if you now link to the alignment, oops. Final tree used.
So return.
Yeah. So it opens something like this, where you can see the tree and the alignment in this format, where you are only seeing the aligned blocks. You can actually change the format of the alignment that you are seeing by saying align type is full sec, for instance. So then you open the tree, you can zoom in, and you have it's interactive also. You can click on nodes, etc., and you have access to the tree. Okay, this is just handy things that you can use always with ET. Next, there's a task here that we already covered. Let's have a look at the tree that we infer with ET view. This is what we just did. Visualize the tree and alignment. We just did this with the ALG and ALG type options. Now, next task in this notebook is generate a new tree using alignment trimming option. So we can modify the workflow. So I'm not going to cover all the examples that are here. Actually, what I want to tell you is that you can use ET build to build many workflows at once. So this is the command that we use to generate one tree, right? So I'm again making this larger. We create a new cell, and I'm going to run the same ET build command, but in this case, I want to use a trimming option. So we don't need to remove this workflow. We can actually provide several workflows. So I'm going to add here another workflow name where this none associated to the trimming is going to be trimal 01, for example, 03, it was said in the example. So we run it, trimal is running. So now it is smart enough to detect that this first workflow was already there. So it's actually not modifying, and it's actually reusing some of the tasks. And in the case of the second workflow, it's going to trim the alignment with trimal and then infer a new tree with fast tree. So it should work. And we have generated like this. So if we now, list, oops, test tree, we have two workflows. And inside each folder, you have a different tree, one without the trimming and one with the trimming, okay? So this is just a very brief introduction to how ET build works. There are some more examples here in the notebook. For instance, this one, we are doing the same, but running 5 different trimming options, workflows, etc. So if you run this, you can try in this workflow, you will see that the same output is completed. It takes a little bit longer because it's running more workflows, but in the end, you are going to have a folder in the test tree where you have all these, all these workflows. We are going to run it in the background. So this is running, okay? We will have something like this, and now you can even use ETView to generate images or even interactively analyze all these trees. So here is just very quickly a quick loop over all the MAFT workflows in our test tree. And we are calling ET3View in this case to visualize the tree, but note that we are using here -E option. -E means that we don't want the GUI to appear and interact with the tree. We want to generate an image out of it. So when this is done, we will have something like this. And if you run this, then there are like 5 or 6 images that are generated, which are the ones that you are using here, that you are seeing here. Let's see if this finished. Okay, we are done there. All the workflows are there, okay? So now if we run this bash loop, ETView is generating an image for each tree in each folder. And this is just a Python snippet to load each image here in the notebook, and you can see. So this is the workflow that we have used. You can see how the trimming is actually affecting the alignment from the trimal 0.3. We are removing some in blocks, etc. Okay, so this is like a crash course of how to use ETBuild to reconstruct phylogenies in a very automated way. And of course, feel free to change any application binding, etc. that you have available in ETBuild. You can even create your own bindings to your own tools if you want. This is ET3 build is just meant to be a wrapper for phylogenetic workflow. So in one line, you can generate everything, meaning that now we can automate everything. So remember that we have all the FASTA files that we generated for all the gene families in our genome, right? And those FASTA files are in data FASTA. We can, with this small piece of code, I'm not going to type it because this is basic. So what I have done is reading the FASTA file and creating a new file that is called target families with the names of all the FASTA files that we want to process, okay? So there should be, so you count here how many lines we have. We have 2,605 FASTA files that we want to process. So phylogenies that we want to build. How to build this is up to you. You can generate one command and submit it to the clusters. You can run it one after the other in your computer. You can parallel with parallel, etc. In this case, we are not going to run this because it takes like half an hour or 1 hour to build all the phylogenies. So this is done. You can automate ETBuild, so to generate an ETBuild command where the input is each of the target families that we have here. So here I'm just echoing the command, but we are not actually running this. The output of this is what you should have in data philo. So if you ls here, you have all the output folders of ETBuild for all the FASTA files. So we have generated all our trees here, okay? And this is our starting point. This is where we can start analyzing from a phylogenomic point of view. We have generated 2,605 phylogenetic trees automatically and very easily with ETBuild, which is the main purpose of ETBuild, automating the process. Okay, I'm going to switch to the second notebook in this category, so building gene phylogenies. We have reconstructed the phylogenies, or how we start analyzing those phylogenies. I'm going to stop 2 minutes. Is there any important questions so far? Everything okay?
One question
Okay.
from Maria.
Maria said, just a quick question for later if you have time. If we want to cluster proteins consider remote homologs, low percentage, blah, blah, blah. What are your two proteins? Could we use MMSEX? Yeah, well, I guess this is a question more for the MMSEX developers. We are actually using MMSEX for clustering remote homologs or relatively remote homologs, and it works relatively well. It's not maybe as sensitive as BLAST, but there are some options that you can activate in MMSEX that should make the job more or less reliable. So if you can afford BLAST or any other method, then go for it. But if you have a lot of sequences, then MM6 is just impressive in terms of time. So we are using it for not 15 genomes, but 1,000 of genomes, and then blast this out of the game. So you have to go for MM6. There's nothing else. Any other questions regarding how we build phylogenes with ETE? I mean, there are many other options, etc. Just check the technical documentation and you will see. For now, we just want to analyze all the trees and show you how to analyze the trees. It doesn't matter if we generated the trees with FastTree or IQ3, etc. This is just up to you. Okay. So if you have generated all the files, all the NEWIC files already, and you have them here, the next thing you need is to actually analyze them in an automated way as well. So this is where the API mode of ETE is taking place. Because so far we have used the command line tools that are very handy to generate data. But when you want to analyze it and more in detail, etc., you need to do it programmatically. So meaning that you want to have your own Python script, load the data, and then interact with the data. So this is where ETE3 package is more powerful because you can actually use it as a Python module. And there are some classes that are facilitating a lot the work with trees in general. So the basic class for interacting with trees in ETE is actually not phylotree, but tree. Tree is basically the same as phylotree, but there are some functionality that it only makes sense if you are working with phylogenetic trees. For instance, considering a species, etc. This doesn't make sense for a clustering tree, for instance. So we are going to be working on the phyllo tree. And the first thing we are going to do is just loading all our phylogenes. So let's do it with some examples. So from ETE3, import phyllo tree. So we have our object here. There we go. Now for any tree name that we have, I call it base name here, in open. Remember that we created a file that it was called target families.txt. So here, each name that I'm loading here is actually the name of one of the phylogenes that we generated. And we know that this is actually stored in data, philo, then we have the base name, and then the workflow that we use. So I'm going to copy this from the other side, that is faster. So this is just creating the path on how to use how to access the file. So these are the paths. Sorry, I have to actually convert base name. So there we go. These are all the files that we want to load, right? So let's check that everything is okay. So if we list this, the path is okay. Oops. Yes. Yeah, we need to clean up base. There we go. Final tree. So let's check one of these. Maybe there's a typo still. What is happening? I don't see it. Ah, it is concatenating twice the.
No, it's okay. You just pasted it wrong.
Yeah. I messed up the. So let's copy paste again. There we go. Okay, so the paths are okay. So and if you actually take a look at this is how the NEWIC format looks like. So this is the parentheses, nested hierarchically, etc. Okay, so now what we are going to do is just not reading the NEWIC, but using phylotree to load each of these paths. And this is our tree. valid syntax. Okay, so this is a quick loop. We have now processed all the trees that we have in the folder philo, and we have actually parsed the tree very quickly with phylotree. It takes nothing, as you see. But we want to store the tree somewhere, right? So this is what we are doing in the notebook, notebook with the solution, with the trees. So we are doing some extra check-ins, etc. But what we are doing is just generating a dictionary that is called all trees, and we are storing the trees there. So all trees base name, which is the name of the tree, equal tree. All trees. There you go. So now we have our dictionary with all the trees there. And the last part that we are doing, I'm going to copy paste, is basically exporting these, because we are going to be using all those trees later on. So I'm exporting the trees, the objects actually, with pickle, which is a Python feature. You can use it to serialize any object. So doing like this, it says, we have to import pickle, and there you go. So we loaded 2,600 trees, and we exported them into data all trees pickle. Okay, so there we go. We have all our trees there. If we need to load them, we will need to use pickle again. Now, there is a trick with phylo-trees that is very handy. Now we have loaded the trees, just plain trees. But if those are phylo-trees and we know that the sequence names are encoded in a species name, we can use ETE to automatically parse these. So you can define a small Python function that is going to be called extract, how I call it here, extract SP code, that is accepting the sequence name, if you want, sec name. And then we know that our sequence names are always sequence dot, and I mean a species code dot sequence name. So I can split by dot and take the first part, and this is the species code, right? So this function is taking any sequence name and returning the species code. So now, oops, we can use phylotree to link this directly. So I'm going to do this larger. Well, actually, yeah. Let's clean up this a little bit. So I think I'm maybe doing very slow. Name. Then we call here fname. Species naming function, and we reference to extract SP code, right? So what we are doing now is the same. We are loading the trees, but we have associated the function to extract the species, meaning that now if we take any tree in the dictionary, I don't remember the name now, but so for instance, oops, this one. So if we access the dictionary like this, T is an object, is an object, a tree object, so we can print it, and there you go. But we can actually call functions like get species, and what you get is already the set of species that are encoded in this tree automatically, because TT is parsing with our function the species codes that are in there. So you can do, I mean, here you have some examples of other stuff that you can do very, very easily. Here, I think we can run it. It should be the same. We loaded the tree. This is just an example of how to load it. And these are some examples of stuff that you can do with the tree. So here we are accessing the species. You can also access the name of the sequences. So if you change here species by leaf names, oops. You have the sequence names, and there are many other stuff that you can do. So each node is associated to different features. So you have name, you have species because it's a phylo tree. You have the dist, attribute. You have the support attribute as well, the branch support. Everything is parsed and encoded as a Python attribute. You can also use the object to locate specific nodes. So if this is our tree, right? So we are printing here in text how you access this node, for instance. So you can call functions like get common ancestor and provide two names, for instance, this and this, and then you get a pointer to this node, and you can interact with internal node as a subtree. So actually, move this. So this is another tree. It's just a pointer to an internal node in a different tree. So this is very powerful because you can interact with the tree, and each node is a different object. You can even start. So T show is going to open. Sorry, is going to open a graphical user interface here where you can interact with the tree, or you can even render the image. You can provide a file name with PNG or PDF, etc. But we are going to use a trick that is inline to actually show it here in the notebook. So you are rendering images for the same. Okay. This is basically the rest of things that I was explaining in this notebook. So far, where we are, and we are closing the first part of the first notebook. We have generated all phylogenies. We have loaded all the phylogenies as Python objects, phyllo trees. We have stored this in a pickle object that we can load later, and we know now how to interact with the trees and how to query different aspects of the tree. There is a lot of information about functions and attributes, etc., that are associated to the tree nodes. You can check this in the technical documentation. For now, I think we just need to have this. Okay. So let's go back to the notebooks, and let's start playing with the trees. Any questions so far? So the idea is that after each notebook, if you have any, even not question, but if you have some idea or stuff that you would like to do and you don't know if ETE can do it, you can ask, and if it is quick, we can also show it to you how to do it. If not,
I have a stupid question, Jaime. What if the species already encoded in the name, or do you have a species slot inside the node object? Or
well, you can hard code the species attribute in the tree, or if you provide the function as we have done here, then it is dynamic. So if you change the name of a node in the tree, then you don't need to change the species. So the species is going to be inferred automatically.
Okay, thank you.
Okay, no more questions. So then we can move to more interesting stuff. So we have all our trees, 2,600 trees. Let's start analyzing them and doing some phylogenomic, typical phylogenomic analysis. The next section is going to be how to generate a species tree. So far we have generated gene trees. There's a question if we could work with other taxonomies other than NCBI. Not at the moment. I mean, not in the same way that we are using NCBI here in ETE, because ETE has a parser and a specific module for NCBI, meaning that you can convert tax IDs, NCBI tax IDs to other information. If you have GTDB tax IDs, Maxime is asking this, then you can parse the species code, etc., in the same way that we are doing. It's just that the species code that you have is not going to be any useful within ETE, because you cannot convert it into other stuff that you can do with NCBI. But we have plans to provide support for GTDB in the next releases. Okay, I was saying the next typical phylogenomic analysis that one could do is how to infer a species tree out of this bunch of gene trees. So there are, okay, another question. When you were building the gene families, does it take the information of the gene coordinates from NCBI? No. I mean, it's not querying the NCBI at all. So what we have done so far is not querying NCBI. We are just using NCBI tax IDs because then later on we can query these tax IDs and get some information, but we don't have the coordinates. No. If you want to include the coordinates, you have to extract this information on your own and maybe call the coordinates as part of the name, so you can parse later, or have this in a separate file, and you can associate features to each node in the tree, and then you can use it. Okay, so species trees. There are many ways to produce species trees, as you know. ETE has some bindings to facilitate the specific supermatrix workflow, meaning that the supermatrix workflow is basically you select some marker genes that are single copy genes that are present in all species, etc. Then you infer alignments for each marker gene, and you can concatenate the alignments and infer a tree with the concatenated alignments. So this is called supermatrix or concatenated trees. And this is a typical analysis that people do in phylogenomics, and it has some workflows and bindings to do this. So if you open the next workflow, identify marker gene family. So as I said, the first thing you need to build a species tree using concatenated alignments is to locate which of our families are actually good marker genes. Good marker genes are basically those that are single copy, as I said, so there are no duplications, and that families or trees that contain all the species. That would be the perfect scenario. So let's try to file good marker genes, single copy and no and covering all the species trees in our set of 2,600 phylogenies. So in this workflow, the first part is basically loading all our phylogenies. Okay, so here we can copy in the blank one. So here we have all our trees loaded. Well, actually here we already had it. So let's start by checking how many taxa we have and creating a set with all the taxa that is in general covered in all our trees. So we load all the trees, and with this code that I just copied here is I'm creating a set where we are going to store all the tax IDs involved in our analysis, and I'm iterating basically over the dictionary of our trees. So the values of the dictionary are the tree objects. And then I'm calling the function getSpecies for each tree and updating the set. And what we have here is basically a set with all the taxa that is covered in all the trees. So we know that we have 15 taxa, and those are the IDs. So this is all the information that we need to start filtering our trees and locating which ones are covering these 15 species and only one copy per species, right? So how would we do that? So first thing, we need to iterate again over all the trees. So the same here for T name and T in all trees items. So we are going to process each tree. And now there is a very quick way to ask this question to the tree. Basically, if getSpecies is the same as getLeafNames, for instance, then we know that there are no duplications. So if the same number of species, if the number of species is the same as the number of genes, then there are no genes duplicated. And also we want that the total number of species is 15, right? So because we counted before, or we could actually check if some specific species are covered there, etc. So this is equal to 15. So if this happens, then we have a marker gene. Let's print it. Okay. So here we are printing the trees that contain only one gene per species and are covering all the species. How many we have? Let's count it. With all species, and we are going to store the tree. Actually, with all SP append and let's store the name of the tree. And then we can print len with all SP. So we have 33 out of 2,600 trees. There are only 33 that have no duplication and cover all the species. Those are the good marker genes that we want to use, right? So this is what we are doing also in this piece of code here in the notebook. Because we don't have the old taxa, yeah, here, yeah. This is okay. This was another question. I solved the question directly here. It is split into two tasks. So the first one is how many trees do contain all the species. So if you see here, we are just checking how many species, how many trees contain the same number of sequences, the same number of, the total number of species. So there are 115 trees that cover all the species, but not all of them are free of duplications. And this is what we are including here, right? Which is what is solved, and I think it's solved also here. Yes, we can rent the tree. Yeah, this is basically the same as before. And here is where we are locating the trees that are good marker genes. I am solving the problem is slightly different here, but it's basically the same code. So we detect 33 good marker genes. Okay. Can we access the tree? Yes, of course. So we have with all species is our list. So now we can say all trees with all species, and we take one random name there. This is actually a tree object. We could have it here, and now we can explore. Show. There you go. So this is our tree, and as you see, it's 15 sequences, no duplications, all the species covered. Okay. So end of this part. We have identified all our marker genes very quickly, processing all the trees, and we have stored the information in a pickle file, which is what we are doing here. So this with all species list is something that we are going to store as species markers. Okay. I'm going to execute this cell. I'm sorry. Species markers, yeah, because we have to execute this. There we go. So now we should have in data. Well, in this notebook, it's not going to be needed, but now we have a file, a pickle file, where we have stored the 33 names of the trees that are good markers. Okay. Very simple. Now we can infer a tree with these 33 markers. Any questions so far? Because the next step is inferring the tree. No questions? Yeah.
I have one question. Why do you require all the species to be present? Can you just consider if a missing gene as a gap or something like this in an alignment?
You could actually infer the species tree with gaps, so you don't need to have the 15 species there. So perfect markers would be those one, single copy orthologs that are also present in all the species. But if you want to work with 90%, like there is a question now by Robert. Can you have more flexibility like present? Yeah, absolutely. For inferring the species tree with ET build, as I will show now, yes, you have this flexibility, and for finding your marker genes is up to you. I mean, when in this code, I'm saying that I want to have 15 species, right? You can change this. You can say, okay, give me all the single copy, but with 10 species, 35. So we included a few more, etc. And you can be more flexible. Only 5, fine. You have more marker genes. It is more important that those genes are single copy, because otherwise then you have more problems to identify which is the best ortholog, and you also have some misleading information from the paralog. So single copy orthologs present in at least 5 species. Yes, you can use it. And then when you infer the concatenated tree, then you need to keep in mind that you are inferring the alignment with gaps. So some families, some markers are going to be only covering 5 species. That's fine. So for the other 10 species, you don't have information for this marker, but maybe other markers are providing the information. You can do it this way. Okay. So next notebook is how we use these marker genes to infer the species tree, the concatenated tree. So you open the 02 infer concatenated species tree in the same folder, and here what we are going to use is again ETE build, but with an additional option that is the supermatrix workflow. So first thing we need in this, what we are doing in this notebook is loading the trees. We don't need this because we already have it. Okay. Well, actually, we need to change this. So we have the 33. Okay. And now, how ETE build is helping you to run the concatenated tree is by reading a file, an additional file that is called the marker COGS. COGS is cluster of orthologous groups. So this is what we have, marker genes that are clusters of orthologous groups. So if you provide this file to ETE, then it will automatically detect which families need to be aligned together and how to concatenate them. So the format of this file, you can check the documentation in the technical documentation of ETE. Okay, I'm not going to explain this, but the marker COGS need to be a text file where each line represents a family, a marker gene. And in each line, there is a tab delimited list of sequence names. This is all you need. So we can generate a file now. This is what we are doing here. Oops, sorry. I'm going to execute it. So we are going to generate a file that is called marker COGS, where we are going to list all the sequence names of each family. So here, this is called with all species. Okay. So basically, I iterate over the name of the tree that is considered a marker. Then I take the tree from our dictionary, and then I access the get leaf names. So this is going to give me a list of sequences in this tree, and I'm just printing them separated by tab, as easy as this. There you go. So now we have a file. Let's check it. That looks like this. Okay. If you count the number of lines, you have 33 because there are 33 marker genes, and we have 1 line per marker gene. So this is all you need to build the concatenated tree. Now we can call ETE build with the additional options. So remember that ETE build for building gene trees, it was using this syntax. So the -W is the workflow that we want to use to build gene trees. We are not needing a full gene tree workflow here because we only need to build alignments for each family, right? We don't need to have a tree for each gene family because we are looking for the final tree. So in this case, we are going to call a gene tree workflow where we only infer a multiple sequence alignment for each family. And the rest of tasks are set to none. Okay, so this is going to read our file, our input, and generate 1 multiple sequence alignment per family. Fine. Now, what do we want to do with those families, with those alignments? We want to concatenate them in a supermatrix manner. So the -M option is calling the supermatrix workflows. And again, here there are many built-in options in ETE to run it. You can define your own, etc. Just check the technical documentation. In our case, because we want this fast, we are going to use a workflow that it has an alias that is fast tree all. So in this case, we are going to infer the final tree with fast tree to make it fast, and all refers exactly to the question that was previously asked. Should we use all the COGS that we have in the file, or can we be flexible? So all means that all the marker genes should have all the species covered. if you define here 90% or 80%, or I don't remember exactly which options are built in, included in ETE, but then you can be more flexible. And if you need to modify this a little bit, just check the documentation of Supermatrix workflow, because you can vary this. The important part here is that we are going to generate multiple sequence alignments per family, and then concatenate them. ETE will concatenate at the species level and infer a tree with all the cogs provided and using a fast tree. How we provide the sequences? Well, we have the -A option to provide the amino acid sequences. You can also provide nucleotide sequences with -M in ETE build, by the way. In our case, we should concatenate all the FASTA files that contain the sequences for these 33 markers, but ETE is smart enough to select from a larger set. So actually, if you provide the all prods dot, this contains all the genomes, right? But it doesn't matter, because ETE is going to read the COGS file and is going to select only the sequences that are needed from this big file. And then after this, you have to provide the COGS file, which is with the COGS option. This is the most important part, because it's going to, data marker COGS, TSV. This is going to provide information of what families to use. And the other key important parameter to run this, and that's why all the formatting advices that I gave you before, it needs a way to know where is the species code in the sequence name. So the same, similar to what we were using with phylotree, we need to provide here. And there is an option called SP name delimiter that we can set it to dot. And then by default, ETE is going to understand that the sequence names are always species dot sequence name. And this is all we need to generate what we have. The other options are actually just technical options. So let's run this. I'm going to say that we use 5 CPUs in my computer, for instance. Oh, sorry, something failed. The output there, of course. So we want this in SP3. Let's go save it in data SP3. So this is running, and you see what we are doing here. I'm going to expand this a little bit. So the first thing it is doing is detecting how many cogs we are going to be using. So here you have how many cogs, 33 are used, are considered single copy, etc., and being used. So 100% of the, so the 33 cogs are covering 100% of the species present in the analysis. Then there is a task called concatenated alignment, and this is running individual alignments for each gene family and putting them together, concatenating by the species code. And when the concatenated alignment is done, it's going to launch the fast tree. This is going to take a little bit longer because it's reconstructing 33 alignments. But in the end, it will finish and provide the final tree. Okay, so it finished all the single gene alignments. It inferred the concatenated, and now it's running the fast tree. It's taking a bit longer because this is a longer multiple sequence alignment. There is meanwhile this is running in the textbook, in the notebook with all the solutions. There is another task that is, well, not this one, using codon alignments. We are not going to cover this because François is touching this step later, but ETE build has the option to run workflows with amino acid sequences or with nucleotide sequences, and it's calling the programs in the correct way. But there is one nice option in ETE that you can use to run the alignment, the initial alignment with amino acids, and then switch back to nucleotides when the amino acid sequences are too similar. And this is a parameter that is called NT switch threshold. So if you say here 0.9 means that whenever amino acid sequences are too similar, more than 90% identity, then it will switch to the nucleotide sequences to infer the tree. If you set it to 0, this is a trick by which you infer the alignments with amino acid, and then all those alignments are converted automatically into codon alignments, into nucleotide versions, okay? It's the same, you just need to call the same way the program, but apart from providing the amino acid, you need to provide the nucleotides and the parameter when you want to switch to nucleotides. So this finished, okay? As you see, we have here the output with the final tree, the path of the final tree. This is our concatenated tree. Let's have a look. The paths are very long, so that's why in this notebook I'm taking this trick that I'm going to reproduce here. So let's copy the final tree into SP3 file. Okay. So we should have now data sp3.niwik, and again this is a niwik format, normal niwik format, and now we can operate with this in the same way as with other phylogenetic trees. But now we can start using some tricks encoded in ETE if you are using NCBI tax IDs. So what we have here actually is, let's do this first. So ET3 view, and we want to check the data SP3 that we just generated. So how it looks, something like this, right? The tax IDs are there. ET build extracted the tax IDs and built this phylogenetic tree. But this is very hard to read unless you know by heart all the tax IDs. So that's why it's very convenient to use NCBI tax IDs with ETE, because you can automatically convert. So there is another tool called Annotate in ET3 that you can use to associate information to the nodes. But there is one special flag that is meant for NCBI trees, let's say. So if you do it like this, you are annotating this niwik file, and you see, I mean, this is hard to read, but this is the same niwik format, but there are some tags here where there is some additional information included. This information is actually all the data associated to the tax IDs, and all the tools in ETE play very well with each other. So you can now visualize, you can pipe this to ETE view and say, hey, this tree contains some NCBI features. Can you show it? Yeah, there you go. So the same tree is already being visualized with the tax IDs resolved and some nice features included here, etc. Interactively or programmatically, as we will see later. So if you click on the nodes, you see that there are some attributes like the lineage, the rank, the scientific name, etc., that are associated there. So this is very cool and very handy if you can work on NCBI tax IDs. I hope in the future also with the GTDB codes. Fine. Can we also operate programmatically with this? Of course, we can. Oh, sorry. We can load with phylo3. So phylo3, and we provide the path. So this is P3. We can print the tree, and we can query stuff like, there are some examples here about distances. So if you want to know the distance between two taxa in this species tree that we just created, you can do it. Oh, sorry. Not found. Yeah, because
SP3, no?
SP3, yeah. So there is 0 distance between this and this. Remember those tax IDs? Probably not, but this is the one that we invented for our strain. It doesn't exist actually, but we know that this 0 distance with this other tax ID, which is actually the other Aquifex. So actually our strain is at least based on the marker genes is identical to the one that is in the reference set. If we want to compute all the distances between all the species in this tree, we can also do it. You have options in each tree or tree node. You can call functions like cophonetic matrix, for instance. So there you go. You have all against all nodes, and you have the branch length distance computing from one to the other. This is just a few examples of things that you can do with the species trees. So fine. I mean, this was very fast, and we have already generated a concatenated tree with 33 good marker genes for extracted from 2,600 phylogeny. So it's very cool what we can do as quick as what you have seen so far. Any questions so far? Because otherwise we move to the next.
I have one question. Sorry.
Yeah, François, you are learning more than the audience.
What is why would you use this threshold option to switch from nucleotides to amino acid?
I couldn't hear you very
this
well.
threshold option that you have, why would you use it in the ETE build? Do you know this?
Yeah, so the idea why I implemented this in ETE build is because when I was analyzing families or species tree, etc., where the sequences are too similar in amino acid, and this is something that happens with marker genes. So remember that if you are building the concatenated alignment of marker genes that are single copy and present in all species are by definition very conserved. So if your sequences are very conserved, probably the amino acid sequence is going to be almost identical between all the taxa that you consider. So the signal that you get in the phylogenetic signal that you get is probably not enough sometimes. And then you want to switch to nucleotides because then you can take into account the non-synonymous, the synonymous mutations. But I wanted to keep the resolution that you get from amino acid based or alignments or codon alignments. Basically, it is always better to align with amino acid. Right, because it's easier, it's technically easier, and the alignments are better. And then if you want to work on nucleotides, you can convert back your amino acid alignment into nucleotides. And then you infer the tree based on nucleotides instead of amino acid. So that's the reason why I implemented this option that is doing this automatically. So you can do it by default always. So you align by amino acid and then you switch to nucleotides, or you only switch if the amino acid sequences in the tree are too identical with each other.
Okay, thank you.
Any other question here, or discussion, or ideas, or I don't know, future implementations that you would like to see in this direction? Can I ask, are you following more or less, or you're completely, because I don't see anybody, I'm talking to a wall. So.
There is one question in the forum from Christina.
From where?
I see you can go from DNA to AA and back, from AA to DNA, and remove stop codons.
Where is this? I cannot see.
It's a discussion forum. If we can go back from amino acid to DNA, so no.
No. And the discussion forum, I think I'm not following this.
Well,
yeah, okay.
On the right.
So if there is something in the discussion forum, you can tell to me. Okay, because I'm not reading very well that.
Okay, I'm answering by chat.
Okay, Christina is Lisa. Okay, she's talking to me from a different chat. Okay, good. So where we are, we have generated gene families. We have built automatically all the phylogenies. We have loaded all the families as ET objects and operated with them, etc. We have located and identified good marker genes, and we have inferred a species tree. So this connects very well with the next module that we wanted to show you, which is about comparing trees. So ET also has options for comparing trees in a very convenient and flexible way. And you may need to compare trees in many different scenarios. One very standard is you have two species trees and you want to compare where they differ, or if they differ at all, etc. So probably you are familiar with the Robinson falls distance, or just counting how many branches are in one tree and not in the other, and all this stuff. So ETE implements these basic distances in both in the API and in the command line tool ETE compare. The basic comparison that other tools can also do is comparing one species tree with 15 species to another species tree with the same species but inferred differently. If you change the size of the tree, for instance, then some programs start giving problems because you need to have exactly the same taxa, etc. And what is even more difficult is to compare gene trees with species trees, right? This is more difficult because the names that you have in the gene trees are sequence names, and the names that you have in the species trees are the taxa. So how you reconcile these is something that you can do very easily with ETE. So I'm going to give you some examples on how to run comparisons in the context of our analysis. So now we identify these marker genes and we invert our species tree. So there is always a question, how many of our marker genes are actually fully congruent with the species trees, with the species tree that we infer. And we can do this by comparing all the trees. So open the next notebook in compare and go to the compare marker families with the species tree. Here, the first part as always is loading the trees. And because we are going to be running comparisons using the first of all, the comparison tool, the ETE compare tool, what we are going to do you can do it in different ways, but to facilitate and to make it cleaner, we are going to take all our species markers that you see here and obtain write the NIWIC file into a separate file. So we want to have a file where each line is a NIWIC tree, okay? And this is going to be our target gene trees that we want to compare. So how you do this? Well, it's very easy. The only interesting part here is this one. So I'm going to copy this part, which is the boring part. So we load the marker genes that we have. Actually, here it was called differently, right? So here it was sp3 is not found. So yeah, with all species, I think. I'm going to change the name just for make it compatible with the other notebook. So there we go. We are accessing each marker tree as before, but now we are going to call this function t3.write. This is a function that we use to export the tree object again as a NIWIC. And we are going to do it like this first to show you something. So if you call twrite and you write it into the file, what you are obtaining is, actually, you can see it here. If you write this, what you get is the NIWIC string. So we are dumping the NIWIC string into this file. So now in this file, if you marker NIWICs, if you take a look, NIWICs, there you go. You have one NIWIC per line, and you should have 33 because we have exported 33 marker trees. So now let's try something. Let's try to compare all those trees, and this is done. I mean, you can check the help of the ETE compare tool, ETE3. So you can provide different inputs. You can compare one tree with another, or a list of trees with another list of trees. So in our case, we have some source tree list, which is our NIWICs that we just exported. And we want to compare it with a reference tree that is the sp3, remember? So we have this in sp3.NIWIC. Just like this, it will compare. Let me make it larger. What is happening here? So this is the different columns are the different distant measures, and this is the effective size. So how many branches or edges are being compared. 0, 0, 0. So there is nothing in common between the gene trees, the marker genes, and the species tree, obviously, because in the gene trees we have sequence names, and in the other we have tax IDs. So how we do this? Actually, we would like to use the species code of all these NIWIC against this. For this, you can use, this is a good example to illustrate how you can export specific features in the NIWIC string of trees. So ET allows to do something like this. Features, and then you provide a list of attributes that you want to export, oh sorry, species. So if you do it like this, then you have the same, oh sorry, species. Let's have a look. Again, wrong shortcut. Data marker NIWICs. So it's again the same NIWIC, but look, we have added, ET has added this special tag where there are some attributes included, and you can add here any attribute. You can export this. Well, distance is already encoded, but the support value or anything that you have associated to the different nodes. Okay, so if you do it like this, then you have the support, etc. And those attributes are also something that you can customize. So it's up to you. The good thing is that we have parsed all our trees as phyllo trees. We have associated the species names, and now we can export back the NIWICs containing this information. So now we can compare with the species information there. By default, it's not using it, but ET compare has this option that is saying, if you want to compare two trees that are not coinciding in names, you can tell me what are the attributes that are coinciding between them. So source tree attribute is actually a species. Probably I used a different source tree attribute. Yeah, I think it's fine. See the problem? Like this. There was a typo. Okay, so now it works. Now we are comparing trees with 15 branches. So it detects that the total number of species is 15. And now we have values for, this is the normalized Robinson fold distance, the Robinson fold distance itself, and some other distance measures that I'm not going to enter here. The interesting part, which is also a usual result in phylogenomics, is that most of the marker genes that we use have a tree that is not congruent with the species tree that we infer. Actually, if you check how many of the gene trees are identical to the species tree, there are very few. So we have 1, 2, and I think there is another here, 4, only 4. So this is just to illustrate how you can compare trees of different kind. I'm not going to enter into more details, but you can compare this that we have done with a species tree and a species. You can do it with anything. Imagine that your gene tree have geo terms annotated in the nodes or something like this, and then you want to compare with a clustering tree of geo terms or stuff like this. So how functions group, etc. So you can use the compare to read the tree, the topology, and then use attributes that are associated to the nodes to make actually the comparison. So it's very, very powerful. And of course, this is powerful, of course, in the command line, but in ET, everything is programmatic. So you can actually do the same programmatically, and then it's much more powerful. So imagine that I ask you, could you give me the specific gene trees that are identical to the species tree? So if you need to parse, the output from ET compare, etc., it's more annoying. If you can program a little Python script, it will be faster. You can access a function in any phyllo tree, actually in any tree in ET, where a function called compare. And compare is actually running all these comparisons, the Robinson folds, the common edges, etc. And you can use the same parameters as we use in the command line. The result in this call is actually a dictionary with all the parameters in a Python dictionary, so you can operate with it. So we can run something like this and list which are actually the gene trees that are identical. So the Robinson falls is equal to 0 in these trees that we have. Okay, so we have identified which are the identical gene trees. You can even go farther and see where are the differences. So this dictionary that is being returned here, so we have C, has this look. So we have the Robinson fold distance, the normalized, the number of edges in one tree but not in the other, etc. But you also get information about what are the common edges, common branches between the two trees, and even the mismatches as well. So how many are in the source, how many is in the reference, how many are in common, how many are mismatches, etc. So you can go very much in depth with this analysis and detect, yes, they are different, but where are they different, etc. And this is more or less what I illustrate in this other notebook, where you can actually count how many branches in the species tree that we inferred are supported by the gene trees. So it's essentially the same example. We are using a counter in this case because we want to count how many edges in the reference tree are supported by gene tree. So this is a counter. We define here this counter. And then what we are doing is updating the counter with the edge that is being observed as a common edge in each comparison. And we can count. We can count how many common edges are for the reference. And then what you get is something like this. So now we print edges. I'm using pretty print just to make it more visual. So this is telling us this partition, this edge, this clade, call it as you want, is supported by the 33 marker genes. So this is actually the split of Aquifax, if you remember the description of the exercise. So this is supported in all the marker genes. This other split is also supported by all the gene trees, but then the numbers are getting down. So there are some partitions, some clades, monophyletic clades that are only supported by 11, and you can use this as a gene tree support, associate to the species tree, visualize, blah, 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 etc. So this is up to you. There are many options. Questions so far? Because there is another section, there is another kind of comparisons that we could do with ETE, which is comparing gene trees versus gene trees, taking into account duplications, etc. But this is, I think this is not necessary at the moment. It's better if we move forward. So I'm going to pass to the next notebook. Questions so far? Because now I'm switching with François. He's going to tell you about two other stuff. Any question, idea, or suggestion so far about these topics? Any signal that you are still live? François, you need to continue now, I think.
I'm alive. Yeah.
So I need to stop sharing, right?
Yes, I guess. I can
stop sharing.
Okay. Okay. You see my screen? Yes. Okay, so I'm going to start over a new notebook because I don't have the one of Jaime. So I've created one empty notebook as Jaime. I will now do this linking trees and alignments notebook, which I already have opened here. It's the same as the GitHub page. And yeah, I hope the font is large enough. So I'm using a new folder for this exercise, this evolve folder. I don't know if you have uncompressed it. So So in this folder, in the data folder, you should have evolve, FASTA, and philo folders. Otherwise, you just uncompress it. It's not here actually. It should be there. Okay, so you should have all these three tar GZ downloaded from GitHub, from the repository, and if you just uncompress evolve tar GZ, you should have this evolve folder, and inside the evolve folder you have this 20-something phylogenies. Okay, each of this one is exactly the same as Jaime did, but I did it with special build parameters. Just to have more packed alignments. I will explain later. So I'm gonna quickly show you first how to add an alignment to the tree. So it's some things that you have already seen with Jaime actually in the beginning, in the first or second notebook. So there are two kinds of trees that you can link to an alignment. So these are the philo tree that Jaime already talked about, and evolve tree. Evolve tree is a tree that is implemented in ETE and is basically used to wrap softwares to do hypothesis testing in evolution and to infer selective, positive selection or relaxation or conservation, negative selection in sequences, in a group of genes. So we can just import them. So the philo tree that you are already familiar with, and the evolve tree. Okay. And philo tree. Okay. So we have these two categories, and we want to select one tree. So this is the tree that I'm going to use in this example, and the next one about hypothesis testing. So to link it, so to load it, so you just, I'm copying and pasting, sorry, from the repo, because it's a long path. So we import this this gene, this family from the evolve folder this time. Okay. This is tools that the set of tools, the pipeline that I use to create this phylogeny. So I used MAFT as Jaime and FastTree also as Jaime. I'm just using trimal. Jaime was not trimming the sequences, and I'm doing a strong trimal option that I will explain a bit after, in order to have very clean alignments. So we load the tree, and I'm missing something. Okay. So I don't know why I was there. I just changed the folder. So I load the tree, and we can just have a look at the tree, just as Jaime did until now. Okay, we have the tree, the phylogeny, and now to link the alignment is very simple. It's tree link from alignment, and you just input the alignment format. There are several that are supported. I don't remember which one. I guess Philip and Pamel. Pamel is the tool that we are going to use next. Also kind of special alignment. There are several that are supported. So we just put the path again to the alignment. So it's the same folder as the tree. And yeah. And if we do exactly the same command as before, the tree render, we should already have pasted the alignment. And this is because this tree is a philo tree, and by default, the layout, the drawing engine, will search for sequences inside each inside each of these nodes. Okay, this works with phyllo trees and evol trees, but with the default trees, it's not going to work. It's not going to have a look at the sequences and use this layout. Each tree object is different. Each of these families of trees objects have different default layouts that can be changed, as you will see with Jaime at the end. So that's it. We have the amino acid sequence there. It's very long, very large, as it was aligned by the MAFT engine and cleaned. So this is for amino acids. You can do the same for nucleotides. So by doing this, I'm linking a new alignment, and it's replacing the one before. Okay, it's overwriting the sequence associated to each node here, and you can see that now it's nucleotide sequences, and it's very long. Okay, so nothing very super nice. You can do so. This was for phyllo trees and for evol trees. Okay, this tree object that is meant to be used only to wrap several tools for hypothesis testing. If we do the same, so I do exactly the same as before. I just replace phyllo tree by evol tree. I load the same tree, and I load the same alignment. Okay, now. Okay, and if I do the rendering, I will obtain the same. Okay, the difference is that this time, so I'm loading these guys that are nucleotides. Okay, I'm loading this exactly the same as this one. This used an alignment and FASTA instead of the final tree, which is amino acids. I'm loading nucleotides, and when I run this, I got amino acids. Okay, because this class of trees does a translation. Okay, so there is this interactive viewer that Jaime showed you already. You can see here, and in this case, when you mouse over the amino acids, you can see the codon. Okay, the column number, the amino acids, the column number, and the codon. Okay, just to visually check your alignment. And so it shows amino acids, but truly what is below are codons, and this is to be used for the analysis that I'm going to show now. As you can see, there are some modifications in this interface, this user interface. On the right, you can choose the models, evolutionary models to run. And if you right-click on the nodes, the leaves of the nodes, you can mark a given node, and you can do a couple of things there. There are specific attributes to the nodes that we are going to see in the next notebook. So that's it for linking alignments to trees. I guess it's okay. It was very simple, and we already saw it. Jaime, at the end, will show. Oh yes, I wanted to show you this, but there's not much time. I think I will skip it. So this is just. If you want to hack this this alignment, there are several ways to do it. You can. So this is a sequence phase. All these images are generated through a sequence phase object class, and you can create your layout that will replace the default phylogeny layout. And inside this layout, you can define sequence phase. And sequence phase, so this is the sequence phase that is actually used usually, but by default, it has several parameters, but you can change them. In this case, I say, for example, that I want columns 10 to 50 and columns 81 to 97 to have a different width, okay? Because I have a very long alignment and I want to focus only on particular parts of the alignments. This is just a very simple example in order to start hacking the sequence. Jaime is going to show much more fancy stuff at the end. I already showed a way to summarize better amino acid sequences because this view is a bit hard when the alignment is very long. Okay, so now we can switch to, I don't know if you have questions. We can switch now to the, sorry, I didn't show you, Sibjal. Screen sharing is too small. Yeah, I don't know what to do. I can press increase there. Is it better like this? Well, let me know. I try to pay attention to the chat. Okay, the next notebook. So this is notebook 6. In this one, so I'm not starting from scratch, just as Jaime, it's a bit long. What I did here was to run ETE build on a set of selected trees, selected by, okay, great. Thanks, Sejan. So what I did is to select the 20 trees that I showed you before because they were small and to go fast. And I use exactly the same as Jaime with the switch threshold 0 to be sure that at the end I have nucleotides and not amino acids, otherwise it's useless. I need the nucleotides to get the codons. Then I have this trimal00 that doesn't exist by default in ETE. I created it. And I created it in this file, this trimal00.config. This is something that Jaime mentioned that is in documentation, and he hasn't showed it. So you just, if you want to hack to change the default parameters of a given tool, like maft or fasttree or whatever, in this case it's trimal, you just create a file, put the name of the file that is going to be used there, as a trimming step. Put the name here that is going to be used there, and put the option that you want to add. In this case, I just added this line, automated one, which is a parameter that takes no arguments. Okay, you just say trimal to automate the trimming, and that's it. Okay, so this is description, etc. This is something that is by default, and then I just added this line. It's very, very simple. So I did this on the 20 plus trees that I selected, and I got this these trees in the evolve folder. I don't know, I'm saying 20 something, I don't remember exactly. 18. Okay, the 18 trees. So I got these 18 trees, and under this path, data evolve. Okay, so this time I'm going to use these two imports. Okay, OS, and this is already imported, but anyway, that's the path where I have the trees. I'm using this in order to have shorter lines, and this function here, well, this loop now is to select the trees. So I missed a step, sorry. This step, I will paste it here. So now with ETE evolve, what we are going to do before this is to run the free branch model. Okay, so this is a very simple model. It's going to compute the selective pressure on each branch of a phylogen. Okay, and so the ETE evolve command to use this is this one. So first, I have this line here so this is what Jaime was saying about the parallel program to parallelize ETE3 to build trees, etc. So I'm using all, this is basically saying, take all the files that ends with this, so all the alignment that I generated in the step before, take all these alignments, take the name of this alignment, and so the family name of the alignment, and then run in parallel with 8 CPUs, ETE3, using the tree and using the alignment.
So this is basically calling ETE as many times as phylogenies you want to test, basically.
Exactly.
And the idea, I don't know if you mentioned, I was out for a few minutes, I don't know if you mentioned, but you are working on the set of 18 trees that you selected because, I mean, the actual goal would be we want to test all the 2,000 phylogenies to see if there is evolutionary selection there, adaptation tests, basically. And you can run this with CodeML, as you probably know, but CodeML is a bit difficult to configure, etc. And the program that Francois created, this ETE, is doing all this stuff automatically, like ETB. So it's a wrapper to CodeML. And what he's doing here is running CodeML on these 18 trees. We could do it for the 2,000, but it will take forever. We need a cluster and everything. So that's why we are focusing only on this 18.
Thank you, Jaime. Yes, CodeML is the longest analysis you can do. It's hours for each anything you do. I put the link in the description about where the CodeML documentation, etc., can be found in case you are interested in the details. Inside ETE3, in the documentation, the links that are above in the tutorial, you can find more detailed description about the models that I use. Basically, these are the models that I use the most, that are implemented in ETE, but there are other options. Okay, so you run this. I'm not going to do it now. It should be not more than 5 minutes, something like this. And then inside the eval folder, you should have in each of these guys, you should have these 3 branch models that are generated with a hash code afterwards. And inside you have all the files generated by CodeML that are going to be parsed now automatically by ETE. Because it's a very long documentation, PAML, and I think that using it through ETE makes things much more easy, much more faster.
Those are, François, those are the file names that PAML is using, or you changed? No, this is exactly the same output as PAML.
This is the output of PAML, and it includes also the input, which has to be 3, TMP, etc. And so this is a config file where the input are 3 and alignment, and then the output names are the output of PAML always. Okay, it's like this. It's very, very hard coded. And yeah, that's it. So it's going to be passed by ETE. So I run this with this command here for the 18 trees, phylogenies, and I run a 3 branch model. What does it look like? So it looks like this. Okay, with the notebook, you always have the spoiler before I'm talking. But yeah, so we can. So yeah, before we go into this, well, perhaps we can just load one to see this. So one, we can just load one of these trees, I guess. Well, let's do this slide because otherwise I will lose too many time. So the first analysis you should do when you do CodeML is to check for saturation of synonymous sites. Okay, because non-synonymous sites evolve slowly, usually, and synonymous sites evolve fast. And as there are only 4 possibilities, if enough time passes, you will not be able to differentiate changes from just random assignation. A nucleotide can be changed to a T and then back to an A, and you cannot say if it's a change or not. So this is called saturation of synonymous sites. Because you see before non-synonymous sites. So to check for this, there is a rule of thumb, and it's that the DS value should not be higher than 5. Okay, so what I'm going to do here is to, among these 18 trees, check which of these trees are good. Okay, meaning that they have a low enough synonymous ratio. So for all the files that I have in my folder, which is this one, the data evol path, I'm gonna check the model that I run. So so there's a loop for each model, but in this case it doesn't really make sense. Anyway, I'm using it to grab the names. It's a bit complicated. So for the free branch model, it will be always free branch model here. And if it's not passing, because there are other files there. Then I load the tree, and I actually link it to the evolutionary model. So by doing this, I'm just saying to ET that under this path, you will have this out file, which is this guy here inside the free branch folder, this out file, which contains the result of panel. So I'm just showing it to you. It's a very, very, very long file where you have a lot of information. Well, not that long actually, but yeah, it's a bit complicated to grab at first. So by doing this, we got the tree. What I can do here is to break and just get the first tree. Check. So I got the tree, and I can use those things. Okay, we got the tree here, and this is how the trees are loaded. So in evol tree, so you have this model that is selected. This is a free branch model that we just ran, and we have these balls that represent the positive selection in the branches that are selected. So here you have a very high omega value. Omega value is dn over ds. So there's much more non-synonymous mutation with respect to synonymous mutation, much more that is expected. This is infinite also there, and this is 19 omega value of 9. This is very, very high. Usually there's a problem with alignment when you have this much high omega values. And for each of the branches, you have this omega value, and you have these features of DN, non-synonymous and synonymous mutations. Okay, so this is what you get if you run it through ETE Codema. So what I'm going to do is what I said is to check that each node, the DS of each node is below 5. Okay. This is what I want to check, and I want to check it for all the nodes in the phylogeny, not only the leaves, but all the nodes in the tree. Okay, so for each descendant of the tree is like the root. Yeah, I will go there. If then that's it. I put the tree there. Okay. I think that's it. Or I put all this. And the fun. Okay. Okay, so I have my good trees there, which are only 2. Not sure why I got less trees. Anyway, so we can have a look at them just as before. We display here in the notebook directly. Good trees, and we have these 2 trees. Okay, I'm missing one. I'm missing my example. Fantastic. So we have these two trees there. And what we can do now is, so this is a general analysis, so only amongst our 18 trees, only 2 passed our filters of DS, below 5. I'm missing something, sorry. So, okay. Anyway, so I'm missing the the file that is my example precisely. I don't know why. Okay, because it does not have the free branch model. Okay, so I will run it quickly there. So I run the free branch here with 1 CPU. I think you should have it in your, well, perhaps it's faster if I,
So you can probably use the one that is in the dark, right? Or
okay, so yeah, I will. Okay, let's load it. So you will have to do it also with me. I don't know why well, I removed it from the. So what we can do is before this, we have the tree. Just check. Okay, this is the tree that I want. We add the sequence. There. And we run the model. So this free branch. Okay, sorry. Just have to set the path of the CodeML, binary, and the path to the output folder. Okay, and now it should work. Okay, so it's 1 minute. I'm just running this this tree. I don't know why I removed it from the results, which is the most important. Okay, so what I'm gonna do next is to take these 3 trees, okay, that have a low DS, and one of them will be my example, and so I will select it. So under tree, etc. So this will do afterwards. Okay, so we'll have the tree there, but it's not necessary now. This is the one that we are running now. And when this is done, link to alignment, this is already done. exact path and work dir. This is also already done. I will paste it here anyway. So basically, we link the tree to the alignment. We define the path of CodeML, where the binary is. By default, if you follow the tutorial, it will be there, which does not correspond to the default of ET because it's a bit outdated. I should change this. But anyway, you can just, it's not long, it's just one line. Just pass the binary. We see the tree. Okay, this is done now. Now we should have.
So, François, just something super basic, but I got a bit lost. So everything that you are doing here programmatically is the same as you could do from the command line, basically. But you want to programmatically to access the all the information and attributes directly there in the script. Because
yes,
it
exactly.
looks much more complicated programmatically. So you actually only need to do this if you need to hack inside the tree and the parameters, etc. Otherwise, I think for most people, just running ETE from the command line will get the results done, and then they can parse the text files, right? Yeah.
So what I did, I have this folder with 18 trees, and from this folder I use ET3eval tool command line. Okay. And now I want to select the one that have a low enough number of synonymous sites in order to be trusted.
So you are parsing everything.
So this parsing, etc., you have to do it programmatically, and the tool is not implemented to check for synonymous sites. And usually well, if you know your phylogeny, you don't really need it. If you work with mammals, etc., you don't need it. I'm doing this because we are working with archaea and bacteria, and the distances can be huge. So this is a check that you should do only if you're working with this kind of species tree, of gene trees.
Remember, we still have time, but it's less than an hour. So
yeah,
there are 3 more notebooks. So just
sorry. So yeah, I will just go through the end very quickly. So I ran this free branch, which should be already run before. I got these 3, and that's it. So this is already run. We have these 3 here. Perhaps it's not linked to the alignment. Well, let's continue on this page. Sorry, it was a bit longer. So here we have 2 nodes with high omegas, etc. Inside the tree, there are nodes, node IDs that are used to label internal nodes by PAML. Well, it's something stupid. So for each node, you can get, as I said, the non-synonymous and synonymous size and the omega values, the ratio, dn over ds. So this is once you know that your tree is okay, you can go on with the analysis and do analysis by site. Okay, you can run this site model that will give you this kind of result. Okay, through ET, it will look like this. It will, the M1 model is a model of relaxation where the omega value is not allowed to be higher than 1, and M2 is a positive selection where the omega value is allowed to be higher than 1. And the whole game will be to know which of the model has a better fit through a likelihood ratio test. And so each of the model will mark given sites saying, okay, this one here are with omega equal to 1, so relaxed sites with no effect of natural selection on them. For the M1 model, and on the M2 model, it will be, okay, these sites are potentially under positive selection, very important potentially for the function. And then what you have to do is to compare which of these models has a better fit. You can tune a bit the display or whatever. This is the most important. You get the most likely model. So you put the most complex model, the one with positive selection, against the null model, the M1 with neutral evolution. And it says that the most likely model is M1, meaning that you don't have per all site positive selection on these 2. Then there are more complex models. To understand fully this, you have to go a bit to the documentation of CodeML, of PAML, and read a couple of papers probably. But this would allow you to quickly see if there is something in your sequences. Okay, so you mark a given tree. This is all programmatically, as Jaime said, and you run a branch site model that will search for positive selection in a given branch in a given site at the same time. So in this case, we mark this branch here as a SUL 102, and we check if a given site in this sequence is evolving at a different rate than the rest of the alignment, the background alignment as it's called. And these are these two lines. On the top, we have M1 and M2 that is from before. And on below, we have the branch site 1, BSA1 and BSA, which is, again, like M2 and M1, one model is going to search for positive selection, allowing omega to be higher than 1. This is the y-axis is the omega value. And the other is for neutrality. And we have one potential site under positive selection, but we have to check which of the model is the fittest. As before, what you can do is a test. So you can just do this command here, just as before. And this is going to work. Yeah, just as before to see, wait, if which one is the fittest. This would be the command. Okay, so in this case, no, there is no positive selection. So all this can be done programmatically. All M1, M2, BSA, BSA1 can be run in one command line, okay, with ETE evolve, very simply. So the tree here, the alignment here, the output folder, the number of CPUs, and the 4 models that I mentioned will be run in parallel, marking the branch of interest. This is the one that we marked before, and that's it. This is only then you get an image after this, which will be the same as the ones that we showed before. This is the image generated by the command line tool. It will also do all the possible tests, all the meaningful tests, comparing the branch sites versus comparing the branch site A and 1 and giving the same results of no positive selection. This is it's a bit hard to see. But this is the model and the alternative and the p-values. You will have also for each site positive selection, etc. I will stop here. I done well. Perhaps you can run this if it's working. So if you run it interactively, you can also mark trees, mark branches in the tree interactively. So in this, I put this interactive flag. In this case, on the same tree, what I want to do is to test if this clade here, so if you click on the tree in the nodes, if this clade is evolving differently from this clade there, okay? So you can mark this tree and also, in order to test this hypothesis, I have to generate another hypothesis, and it's that check. Ah, no, sorry. Is that all these ones here are evolving together. I put these 2 here, and before I put the 3, because this is a node that I don't want to consider in this comparison. So you have the background here, and one I don't consider, and that's it. So by doing this, okay, now I close because I don't want to mark anything more. ETE will run these models with this clade definition, and we'll do the tests. Okay, and we can give you if they are significantly, which is the best model significantly. So yeah, that's it. I'm done for this part. Sorry, Jaime, I was wrong. Do you have any questions? I'm forgetting about the chat. Okay, Jaime, I'm done. Huh? Yes, exactly. That's well, the best way to mask it is to use trimal. Okay, at the beginning, you just this is why I insisted in this part. You have to really be strong on trimming the alignment. I don't think on the CodeML part, there is a way to mark columns in the alignment. You just remove them either manually or trim. You can, as I just did, you can just mark some lines of the alignment in order to assign them a different evolutionary model just for them, as I did for this the gene. That I did it actually because it's an horizontal gene transfer, and it's not a good idea perhaps to consider it with the others. Yes, it's true. In the trimal output, there's a file that is generated automatically where you have the transformation. So it's confusing. There is no way now through the tools. I think, Jaime, I don't know, correct me if I'm wrong. There is no way to do this automatically to the alignment. There's no. This is not kept inside the alignment object. It should be, but it's not done. So programmatically, you have to go through this. The same if you want to map the selective pressures onto the 3D object of your protein. I saw that there's a tutorial about this. If you want to do this, then you should carefully map using trimal. The trimal output, you should carefully map the coordinates between the output of CodeML and the original alignment that you used using this trimal.
Yeah, you're right. So you're finished, François?
Yeah.
And somebody was telling me that there are some questions somewhere in the platform, but I don't see anything. I just, the current questions panel for me is empty. I don't know if you can see them or.
There are questions, the discussion forums that I'm just answered.
I don't see any, so maybe it's my panel or something that is not updating or. Okay, so
no? You don't see this?
Well, I cannot read, but there were some questions about, Anna told me that there are some questions about the domains and the marker Newix txt file, etc., that I cannot see. So maybe I'm going to continue with the notebooks because we are running out of time. But at the end, we can maybe try, if you collect all those questions, at the end, we can try to discuss and solve them. Okay, so I'm sharing the screen again. So I hope you are still there. We know that this is being intense, but we wanted to give you, this is going to be recorded. So you can check later, go back to the parts that you got lost, etc. I think it's better that all the information is there, and then you can work offline as well. But all the features are covered. So we still have 1, 2, 3, 4 notebooks, but actually this one we are not going to touch. So it's 3 more notebooks covering 3 important sections or type of analysis. So wrapping up, we have all our trees iterated out of the FAST file and our gene families. We were building concatenated trees out of them. We were identifying marker genes, etc. We were comparing gene trees with species trees, etc. We know how to link trees and alignments and work on analysis that are combining both, like testing adaptation, etc. So there is one more topic that is very important when you are analyzing data from a phylogenomic point of view, which is making sense of the evolutionary events that the phylogenies are providing you, the topology of the tree is providing you. So this is what we are going to cover very quickly in the next block, the predicting evol events. So the first part is more about duplications, evolutionary events called duplications, and the second is about the speciation. So as you know, and this is super quick explanation, but if you are building a phylogenetic tree and you are connecting different gene sequences, what you are doing is inferring the evolution of all those sequences. And the internal nodes represent like ancestral states in the evolution of those sequences. And also those nodes represent duplication events or speciation events, depending on if the two sequences that are connected to the the same node belong to the same species, then you have a duplication. It's a duplication of the gene in the same genome. But if two genes are separated by, I mean, the two genes are from two different species, then they are separated by a speciation event. And inferring this information out of the trees is crucial, because you treat differently genes that relate by duplication events and genes that relate by speciation events. In the case of speciation events, you have genes that relate by an orthology relationship, and in the case of duplications, you have paralogs. So we really want to extract all this information from all our trees. So we need to go programmatically and load all our trees and start analyzing every internal node in the tree to know if this is a duplication or a speciation. And there is a lot of literature and methods how to do this. We are going to cover something very simple, but still very useful. I mean, this is perfectly accurate for research intentions. So in this notebook, the first thing we are doing is loading the trees. You see, we have our 2,600 trees here, and we are setting some basic tasks that we need to cover with, that we need to do before we analyze the tree, that we interpret the tree. So as you also probably know, all the tree inference programs, or most of them, return a tree that is unrooted. IQ-tree, RAXML, FastTree, all those programs return a tree that is unrooted by default. Even if you see them and they look like rooted, they are not rooted. So the first thing you need to do with a phylogenetic tree, if you want to analyze it, is rooting the tree. This is setting the correct outgroup, so then all the sequences are polarized according to the outgroup, and then you can interpret the internal nodes from an evolutionary perspective. So to do this programmatically, ETE has a function associated to every tree that is set outgroup, this one that you see here. And you can select whatever node that is included in the tree as an outgroup. There are many methods to detect which is the best outgroup. We are going to use the most simple one, also very common, which is getting the midpoint between all the branches in the tree. So ETE also implements this algorithm, so you can call the midpoint, get midpoint outgroup. This is going to give you a pointer of the node that splits the branches into two well-balanced branches, edges, and then set this node as the outgroup of the tree. I'm going to reproduce all this stuff here. So we have all our trees, and first thing we need is to root all the trees to start analyze them. So T name and T in all trees. I think it was called the same here. Items. Well, basically I can copy paste this. So this is, if we run something like this, we are just basically loading the trees and rooting. Actually, the root is already set up. If I rerun this, I'm doing something redundant, because the tree object is already rooted. Once you do it, once you call this function, the tree object is modified. So actually the tree is now rooted, and you don't need to call it again. Then once you have rooted the tree, we can interpret the internal nodes as duplication or speciation. So what is the most simplistic definition of a duplication event? Where, well, a node in the tree that is separating two clades where there is an overlap in a species, right? So there is an algorithm, very simple and but also very effective, that is called species overlap algorithm, that is basically scanning the tree, all the nodes in the tree, and labeling each node as duplication or as speciation, depending on the child branches. If they share a species, this is a duplication. If they don't share a species, this is an speciation. So ETE has this algorithm implemented by default. There is another one more less used but more theoretical, like reconciliation, full reconciliation without any tricks. But we are going to use this because it is a bit more flexible to, or a bit more resilience to artifacts in the trees. So this method, you can check the technical documentation to read the details, but basically you can call it for the root for each tree, and automatically all the nodes are going to be labeled. So we do it like this, and what is going to happen? Oh, one error. This is an error that I wanted to show you. That's why I made it fail. To run the species overlap algorithm, the trees need to be completely bifurcated, meaning that the nodes that have more than 2 childs are not possible to be analyzed with this, at least with this implementation. So you need to check for multifurcations. Depending on the program that you are using to infer the tree, you will get multifurcations or not. But this is very stochastic. Sometimes, I mean, FastTree, for instance, it infers multifurcations when the sequences are identical, for instance, instead of having 0 branch distance length bifurcated topologies, then you have a multifurcation of 3 or 4 branches. And this plays badly with the algorithm. So in ETE, there is one function called standardize. Standardize. That is doing precisely this, plus some other stuff. So standardize is basically converting your NIWIC into something more canonical, like it converts all the multifurcation in bifurcations, but bifurcations with zero branch, so the distance between all the nodes is still 0, and it also solves some technical problems that sometimes are associated with the NIWIC output by some programs. So now it works. So now we can process every tree. We set the outgroup to the midpoint in this case, and we infer the evolutionary events. Events, this thing that we are obtaining here is just a list of evolutionary events, each one pointing to one node in the tree. You can operate with these evolutionary events if you like, but in the end, it is always more, well, not always, but in our case, it's going to be more convenient to operate directly with the tree and check, actually verify that every node in the tree is actually labeled as a speciation or as a duplication node. So let's take one example. We have processed all our trees, so we can access this. This is a random one, and render. So if you see now, the tree image is no longer all black. Now there are blue and red nodes. This is because the nodes in the tree are automatically labeled as duplication and speciation, and ETE by default understands this language. So we know that these blue nodes are speciation events and these red nodes are duplication. So we know that this is a duplication where actually you can see two groups of orthologs, right? So this was a duplication at the base of these 2 species. Cool. So we have the tree already labeled. How this looks programmatically? Well, if you go to something like, this is a way of accessing the children. So this is a children node, and evol type, I think. Yeah. So the first branch after the tree root is duplication. Okay. And the other is duplication as well, and so far so on. Okay, some will be S, some will be D, but that means that we can also operate with the tree and know where we are dealing with a duplication or a speciation. And this is very powerful when you are analyzing phylogenomic data. For instance, it is very common that you want to count how many duplication events happen per tree, or how many duplications happen in one specific lineage, or stuff like this. We can do it. We can do all this programmatically. For instance, there is this task proposed in the notebook. How many duplications events are per tree, and which is the most duplicated family? So which is the most duplicated tree? The code, as you see, is very simple. We are going to analyze each tree. So this is the same loop as always, but in this case, we are going to work with internal nodes. So ETE has this function called traverse that is going to iterate over all nodes, not only leaves. By default, if you do it like this, then you iterate over the leaves of the tree. But now we want to iterate over all the nodes. So we use traverse. And when we are traversing the tree, we can check if the node, the variable n, has the able type label. And we can check if this able type is a D, then we know that this node has a duplication, right? And what we are doing here is just counting how many duplications are per tree. So we can have a counter, and this is what we have here. So I want to count how many duplication events are in each tree. So I'm processing one tree here, I traverse all the nodes, and for every node that is a duplication, I increase the counter with the name of the tree. So every time I see a duplication in T name, I increase the counter. What we have in the end is something like this. So as easy as this, you know which is the most duplicated family in our set, this one. There are 153 duplication events in this family. It's huge. So this is probably ABC transporters or something like this, where there are many in-paralogs and internal duplications, etc. Actually, we can check. So if we want to take a look at one of these tree, we can all trees and then check for this name. Oh, sorry. Copy paste this. And we show as easy as this. There you go. Huge tree, right? And you see so many red branches. These are internal duplications. And actually, yes, there are so many duplications. If you see some of these red blocks that are over there, you see there are many species-specific duplication, lineages, etc. So we have identified very quickly the most duplicated family. And also we have a number of duplications. So we can profile our set of phylogenetic trees to know how many duplications happen in each tree. And we can even go farther and measure how many duplications are being identified per clade. Remember that we were in the reference tree, we could count how many gene trees are supporting each clade. So we could do the same here. We can, when we are in a node that is labeled as a duplication, we can check what are the species involved in this duplication, these ones. So we can count how many duplications happen at each position in the reference tree. Cool. So continuing with our example, now that we know how to operate with duplication events, there is a typical question and check that you will need to do when you are doing phylogenomics, which is, for instance, checking one specific type of duplications, which are lineage-specific duplications. We want to know, remember the description of the problem. We have our Aquifax strain, and we want to check if there is something special there. So why don't we check if there are some specific duplications happening in our proteome? So let's first count how many trees contain our species and our species involved in a duplication event. So this is the tax ID of the reference Aquifax, right? And what I'm doing here is exactly the same as before. I check all the trees, I traverse all the nodes in each tree. If the node is a duplication, then I want to check, and I check, are the species under the duplication event involving any Aquifax sequence? If so, show me. So there are no, well, actually we are not checking if Aquifax is part of the duplication. We are checking if Aquifax is the only species involved in the duplication. That's why it's lineage specific, sorry. So we are checking under this node that we have labeled as a duplication, is there any other species apart from the Aquifax? And the answer is no. So all the Aquifax genes are single copy with respect with itself. So there are no species specific duplications here. Can we repeat with our problem genome, which, remember, it was this invented tax ID. Here something happened. So in this case, what we have identified is some trees. Actually, this is not ref Aquifax anymore. This is our strain. So we have identified some duplication nodes that, yes, I mean, you see the tax ID here. This is obviously a duplication event happening only in the genome of this aquifex. And the cool stuff is that we also identify that this is a difference compared with the reference one. So this could actually be related to the biology, the special biology that we identified in our experiments. So this duplication is very interesting. Actually, I'm not going to do this, but if you continue the exercise a little bit and check what are the sequences associated to this, you will see that actually this is an oxidase, a sulfur oxidase. So there is a duplication specifically in a gene that is very involved in sulfur metabolism. So this has a lot to do with the observation, the experimental observation that we had. And this is how we identified. The type of things that you can do with duplications is endless. You can go with any kind of analysis, and this is up to you basically. All you need to know is that you can operate with the trees in this way, so you know precisely where are the duplications and where are the speciation. The next thing we can do is, instead of focusing on duplication, we can focus on speciation events. So let me go to the next notebook. And this is identifying speciation events, is identifying or inferring orthology relationships based on the tree. So same procedure as before. We need to load all the trees. We standardize because we did it with the fast tree. We root it and we infer the duplication event, the evolutionary events. But in this case, we want to check for speciation events. So if I want to infer the orthology relationships in each tree and have a collection, a table of all the orthology relationships of all species, what should I do? Well, something very, very similar to what we were doing before. We traverse every tree. We traverse we iterate over every tree, and then we traverse the tree. But instead of focusing on nodes that are labeled as duplication, we focus on nodes labeled as speciation. And here, the speciation node is telling us whatever that is in the left side is an ortholog to whatever that is in the right side. And we can extract this information from the tree very easily. So this is what I'm doing here. Side 1, side 2. And we access this with the attribute children. So we know that there is a children on the left and children in the right. And we have this side 1 and side 2 are two internal nodes in the tree. And we know that whatever that is inside 2 is ortholog to whatever that is inside 1. So if we want to infer orthologs, all we need to do is to print all the sequences that are in seed 1 and side 1 and in side 2. So if I do something like this, side 1, get leaf names, and something like this, what I have is all the orthology relationships in all our trees. So we know that this is an ortholog to this, and also this is an ortholog to this, and also this is an ortholog to this, and so on and so forth. And you can organize the output as you want. You can, for instance, something very common is that you just want to know the orthology relationship of a specific species, not all of them. So that's why we are adding this piece of code here. So imagine that your task is to identify what are the orthologs of your strain genome with the reference genome. So in this case, we just need to filter what are the species, speciation events that contain these two species that we are interested. Just one important consideration here. The order in which side 1 and side 2 is organized in the tree is completely random. So you don't know. You just know that these two are random, are orthologous sequences, but it could be the other way around, and the same interpretation stays. So we need to check. We don't know what the species are here and what the species are here. So if we want events when we want aquifex 1 here and aquifex 2 here, we also have to check for the opposite. I hope you are seeing my hand because I'm pointing. So we also want to check the opposite, is aquifex 2, aquifex 1, or aquifex 1, aquifex 2, right? So this is basically what I'm doing here. I'm switching the sides depending where is each tax ID, okay? And I organize this. And then the check is very easy. So if I want to infer only the orthologies of every aquifex, my strain aquifex. With the rest, I select the side, which is the first side, because I organize this, the side of the speciation event only if it contains the aquifex, and whatever that is on the other side is considered an ortholog, right? So now I can print aquifex versus other. And what we are going to get is just well, not with other species. Other species in this case is only the target species, so it's only the other aquifex. If we remove this by another tax ID, then we are inferring all the orthology of aquifex, our strain, with the other genome, and this is our table. We have, as easy as this, all our orthology predictions based on the tree, so fine-grained orthology predictions. Actually, if you check the list, you will see that in most cases it's a one-to-one relationship, right? But there are some interesting ones, like this one, where there are two aquifex, our strain genes that are orthologs at the same time to this. This is a many-to-one orthology relationship. And how to parse this is just a Python question, okay? So if this is one-to-one, then you only need to have one sequence here, one sequence here. If you have more, you are in a many-to-many or one-to-many, etc. So this is how you infer orthology. There were some tasks that I was thinking that we could cover, but I'm going to leave it for now because we are very tight on time. Actually, very tight. It was only 15 minutes. So, and there are two notebooks that I want to check to show you. Yeah. So one more that I think it will be very interesting for you, which is linking to the NCBI taxonomy. So we have seen this already with the ETE NCBI query, etc. command. I'm not going to reproduce everything very in detail for the sake of time, but I want to show you how you can use the NCBI module in ETE to automatically convert your tax IDs into relevant information. So we saw this with the species tree. Remember, we inferred the species tree with tax IDs, and then there was this command annotate that we used to go from a tree with tax IDs to something like this. Okay, so this that we are doing automatically, we can also do it programmatically. And remember that there is also a tool, a command line tool called ET3 NCBI query, where you can also do some quick checks on the terminal. So imagine, so this code from here is just getting the list of taxa that we have in our analysis, right? So if we cut this target taxa, this tax IDs through the command NCBI query, and we say we want the info of this tax ID, then what you get is something like this. Tax ID and the scientific name, the rank, the lineage, etc. Or if you want to have the topology in the NCBI taxonomy that is relating all those taxa, we can do the same. We cut the target taxa. We are going to remove our invented tax ID because it's obviously not in NCBI. And then we, instead of info, we request for the tree to the NCBI module, and we visualize, and where is this? We are generating an image. Sorry, it's here. So if we remove this, what we are getting is the topology. This is not the species tree that we infer. This is the topology that is present in the NCBI taxonomy. Okay. So how we use this programmatically? Well, remember that we were loading all the trees with the tax ID encoded as part of the sequence name. So now it's super easy because the phylotree object has one function called annotate NCBI taxa. So this function NCBI taxa is going to read the species attribute of every node and translate it into meaningful information. So if we do something like this, oh, not here. Our tree already contains three, same, children, .0. So we have, apart from the able type, distance, etc., we have the the name lineage, the scientific name, the rank, etc. All this is present in every node. So now we can use it, right? Yeah, this is the same you are seeing here. You can traverse the tree. Sorry, yes, I didn't load the trees. Yes, and now we can traverse the trees and print tax ID and all the information. If we want to have information about the internal nodes, there is one trick that I'm showing here, but it's not super important, which is that you can modify the function that you are using to return the species code. So we can do the trick that if this tax ID is the invented one, then fake it and use it and return the Equifex one. And then everything is going to play well because all the tax IDs are found. This is just a trick. Where else I had this? I think this is all clear. You can remember that you can export once you have annotated with NCBI or with whatever other features your tree. You can export the NIWIC using features. And if you use this trick, features empty list, all the features are going to be outputted. Okay. So if you do like this, then this test tree NIWIC is going to contain all the features that we have been annotating so far. Okay. Okay, last 10 minutes or so, because I think this is automatically closing, so I don't want to be cut. It's about visualization, which is an important part, a very important part in ETE. And the options are very large. So the most important thing here is the understanding the design of the drawing engine in ETE. So in ETE, there are 3 layers that you can use to control the visualization of trees. And this applies to the interactive GUI, to the rendering, to whatever. So there is the tree style that controls how the tree is going to look, if it is rectangular, circular, using semi-arcs, whatever. Node style, which controls the specific aspect of each node in the tree, color, size, etc. And faces and layout function. Faces are basically small graphical items that you can associate to any node. And layout functions are basically dynamic Python functions that you are using to decide what is the style and what are the faces are associated to each node. Okay, there is a lot of documentation that you can check here and learn how to do. And the other important part to understand how ETE works in visualization is that when you add graphical elements to any node, you can add this to different relative positions in the node. So you can add graphical elements to branch top, to branch bottom, to branch right, floating, branch right, as I said, or align position. And controlling this, you can add as many elements that you want to any node and to any position, and they will stack. If you add 10 elements, then they are going to stack in the align position. So imagine each position as a table. So you have columns and you decide in which column you are adding each element. So I don't know. It's a pity that we don't have more time to play with the visualization, but I added here an example on how you can customize your images a lot. So to make it very quick, and then you can play later, or even if this finishes in 10 minutes, and you want to ask later or whatever, we are going to be available by email or even in the Gitter chat, etc. But basically, we are going to load a tree. Cool. We have the tree, and we are going to create a function that is going to annotate the tree as we want. So this is a function rendering tree that we are doing all the things that we have learned so far, standardizing, midpoint, etc. We are labeling the evolutionary events, we are annotating the NCBI taxa, and we are linking the alignments, okay? And all these, remember, when you are annotating the trees, is creating attributes in every node in the tree. So now you can access the attributes in the node to decide how they are going to look, right? So the first thing we are going to do is create an empty tree style. Here, we can set some title, or even if you want to change some other attributes in the tree style, you can. And the important part here is the tree style has an option that is the layout function. And we are going to say, use our layout function, okay, before rendering, because the next line is rendering. Use our layout function. And our layout function is just a Python function that we define, and we can do whatever we want with the nodes. And this is going to be executed at runtime. So every time a node is going to be visualized, it will pass through this function. So then imagine a Python function that is receiving a node object as a first argument, like here. And now we can decide what to do with the node. So every time a node is going to be rendered, I receive it here. And I say, okay, the default style for this is going to be shape square and black color. Okay. Now, if the node is leaf, then I want to color by able event, right? So this is the same that we were seeing before. If the able type is a duplication, I want it red. If the able type is a speciation, I want it blue. But I want to have some more additional information. I want to have the support values, for instance, of internal nodes. So I can create a phase. A phase, remember, that is one of these graphical elements, which is a text phase. And I can add any text. In this case, it's a text that it also reads from an attribute in the nodes, the support. So I create a text phase that is reading the support and rendering the support with this format. I want it in this size and I want this color. And now I say in the layout function, I use this function that you can import from the ETE Toolkit. And I say, I want this phase to be added to this specific node that I'm analyzing in position branch top column 0. Cool, it will be added there. If it is a leaf, then I want to add more information. I want to, for instance, have the species encoded by the sequence. So we can remember that we have the annotations from NCBI, so we can access node scientific name. So let's create another text phase that is reading from the scientific name and using this color, and we add it. But in this case, we add it to the position align. So it's going to be placed aligned in the right side of the tree. For every internal node, we can also do some fancy checking. For instance, we want to label what are sequences coming from archaea and which ones are bacteria. So we can check from the attribute that is also annotated, name lineage, if archaea is there. So if this sequence that I'm plotting at this moment is an archaea, then use a text phase with the name archaea and put it in a line position. But look that I'm using again a line. So I want it in a different column. I don't want the phases to be stacked. So I'm using column 1. So first we will have the species name, then the label, if it is archaea or bacteria. And then we want to have the alignment. So remember that we can also link to alignments and we can plot it. So there is a special phase in ETE, also built in, that you can import from here, where you can read the attribute in the node, which is the sequence. So sec motif phase, and you provide the sequence, and you say how the sequence should be plotted. In this case, we are going to use compact sec. It could be full sec or blocks or whatever you want. And we add it to the align position number 2. And then finally, for every node in the tree, we want to have the distance also plotted. So we read the attribute distance, we format it, and we add it. And in this case, we are going to add it to branch top, position 0. So branch top, we used it before, is here, and column 0. So they are going to be stacked one after the other. Okay, let's execute this, and it fails. all trees, we don't have them. So we load the trees. Now, visualize, and voila! This is our customized tree. So these are the phases that we have been adding. If it is an internal node, we have support and branch distance. If it is a leaf node, we have only the distance, but plus the species name, the label if it is bacteria or archaea, and even the representation of the aligned sequence. And everything for every node, and it's processed automatically. You can even modify the style of a specific nodes. So you can, without the layout function. So you can access, for instance, the ancestor, the common ancestor, the node that is connecting these two sequences. You get a pointer to the node and you modify the background, this pink. That's why this is being rendered pink. And you can even display partial trees. So in this case, expansion is just a pointer to the common ancestor of these two, right? So we can render this, and the rest of the tree is not rendered. It's just this part. And you can control, I mean, you can go crazy with this. You can modify as much as you want. The cool stuff is that everything is so programmatic, especially if you create it like this in a function, that you can now recycle. We have 2,000 trees in our dictionary and a function, render tree, that we can use to render trees. So we can pick any name, pass it through the render tree function, and we get our image with all the features that we want to analyze, etc. And even more, we can do the same with show. Sorry, show. Well, sorry, I cannot do it this. I should do it in the function. So instead of render here, you can call show, and it will be interactive. Okay. So I'm very sorry that we don't have more time to play with the visualization. For the full day tutorial that we had planned, we were considering much more time for visualization and playing with you, and even having fun with some fancy visualization, but we don't have time for now. So I hope at least it's useful and clear, and you can go from here to any other customization that you would like to do with your own data. We only have 2 minutes. If there are some quick questions or feedback or whatever that you want to express, now is the time. Otherwise, I hope it was very useful, and we can discuss by email or any other chat platform. François, I don't know if you want to say anything else or?
No. Yeah, we don't have time. So if you have questions by email or what you just said.
Yeah, the Gitter chat, we don't use it very much, but if you name us there, we will see it. Also check the GitHub account, where we usually take care of issues, sometimes support questions, etc. And otherwise, just write an email. We are available.
So I found the questions that we were looking for before in the live question and answers. So one is, how do ETE deals with negative branch length?
ETE with negative branch lengths?
Yeah. How does it deal with this kind of problem?
ETE build or ETE div?
ETE. How does ETE deals with?
Negative. It is stored as a negative branch. So when you upload, the only effect is that when you plot it, the line is going to be rendered on the other side. But apart from this, there are no problems.
Okay. I'm not sure if currently there are differences in tip levels among trees. How does ETE handles missing species names in tip levels? An error, no? If one of the nodes doesn't have a species label.
So that this was covered in one of the things one of the tricks I mentioned. So you can fake the tax ID, or otherwise it's going to be just empty information. When you link it to NCBI, if the tax ID is not valid, then there will be no information for that node. And 10 seconds to go. Sorry, this is a bit stressing. Okay, see you guys. I hope it was very useful. Ciao.